Hello students, welcome back to video solutions of part test 4. So in this video, we are going to discuss about different solutions of part test 4. Okay, so before starting, I am Ronia, your botany faculty at MEMONEET. I am currently a final year medical student at Government Medical College, Mehboob-Nagar. Okay, I'm here to clear your all doubts in botany. If you have any doubt, do not hesitate and mention them in comments. Okay, now before starting this video, I want to give you a small tip, okay, regarding NEET. So when I was writing NEET 2019, I used to do one thing. After writing tests, I used to write all my mistakes, whatever the mistake I have done in that test in one notebook, and I called it a mistake notebook. And before writing next examination, I used to read those mistakes so that I do not repeat those mistakes. Okay, because making a mistake again is a big blunder than making a new mistake. Okay, it is good to make new mistake because you will learn to not make that mistake again. But if you repeat making same mistake, it will lead only to reduction of your marks. हाँ, मार्क्स घट जाएंगे. इससे बड़ी प्रॉब्लम और कोई हो ही नहीं सकती है. एक ही गलती को बार बार दोहराना बहुत ही बड़ी ब्लंडर वाला काम है. उसको कभी करना नहीं चाहिए. सो टू अवॉइड दिस थिंग, एक काम कर सकते हैं, एक मिस्टेक नोटबुक बनाइए. जो भी आप मिस्टेक करते हैं एक एग्जामिनेशन में, उसको आप उस नोटबुक में लिखिए और अगले एग्जाम से पहले उसको पढ़िए ताकि वो सेम मिस्टेक दूसरे एग्जाम में रिपीट ना हो. ओके, एंड ऑल्सो आई वॉन्ट टू टेल यू one thing: read common, revise common things will be asked in examination and you will land a medical seat. Understood? Now moving on, let's start with the solution. So to start with question, I'm sorry, this is unread a lot. I'll just erase it. Just kidding. Yes, so question 101. What is question 101 saying? It is saying match the following: scavengers, detritivores, parasite, bacteria, fungi, vulture, crow, termites, and earthworm. We have following. Okay, so scavengers are organisms which feed on dead organisms. Okay, so example for these are vultures and crows. So the option B is 1. Parasites are organisms which feed on other organisms, which reside on other organisms and take nutrition from those organisms. Okay, so example for parasites are bacteria and fungi. Parasites reside on other organisms and take nutrition also from them. What are detrivores? Detrivores are organisms which feed on detritus. Okay, so answer for question number, option 2 is C. So we have answer for option 2 is C. So we have 1B, 2C, A, 3A. 1B, 2C, 3A. So answer would be second option. Okay, let us move on to 102 question. What is this? What it is saying? It is saying given below are two statements. Vertical distribution of species occupying different levels is called stratification. Yes, this is a correct statement. In an ecosystem, there are different types of species. And these species are, when these species are arranged vertically in different levels, one after other, like level 1, level 2, level 3, in an ecosystem, when different species are arranged at different levels, one after other, this arrangement is known as stratification. Okay, so this is a correct statement. After this, what it is saying? It is saying forest, river, grassland, and estuary are some examples of terrestrial ecosystem. See, I would say this is a wrong statement because estuary, my friends, is an example of aquatic ecosystem. Okay, it is an example of aquatic ecosystem. Understood? It is an example of aquatic ecosystem. So this is a wrong statement. So answer would be statement 1 is correct and statement 2 is correct. That is, statement 2 is incorrect. That is option 2. Okay, moving on to next question. I hope this is clear to you. It was a very simple question. I would rate it as 3 out of 4. What is question 103 saying? It is saying the mass of living material at trophic level at a particular time is called standing crop. Okay, so standing crop is nothing but biomass or living material that is present at a particular trophic level at a particular time. What is a trophic level? Trophic level is one level in a food chain. Okay, so there is a food chain. Food chain includes different levels at different levels, and at each level some type of organisms are included. Okay, and at each level some types of organisms are included. The base level always includes organisms which feed on grass and all. So the base layer of the food chain includes organisms which feed on grass, that is herbivorous, and the top layer includes carnivorous. And each level of a food chain is trophic level. Okay, so amount of biomass or living material that is present in this trophic level at a particular time is known as standing crop. And this is the answer to question 103. Let us see other options: net primary productivity, gross primary productivity, and standing state. What is standing state? Standing state is nothing but a functional characteristic of an ecosystem, which includes different parameters like population size, species, etc. So this is characteristic of an ecosystem, functional characteristic of an ecosystem, which includes different parameters like species, population size, etc. Now, I'll tell you one thing, one small equation. Net primary productivity refers to total amount of energy captured and stored by photosynthesis. Okay, by plants, total amount of energy captured and stored by photosynthesis by plants minus energy required by plants for respiration. Okay, so this is net primary productivity. And this total amount of energy captured and stored by photosynthesis by plants is gross primary productivity. I hope it is clear to you. So we have something like NPP is equal to GPP minus energy required for metabolism and or respiration. Okay. Understood? So answer to question 103 is option 2. What is the answer to question 104? It is giving again two statements. This first statement is saying in pyramid of energy, the base of pyramid is broad and it narrows down at the apex. This is a correct statement. So this is the pyramid of energy. The base of pyramid of energy includes producers, that is organisms which produce energy. And as we move up, there is consumption of energy occurring. Therefore, there is decrease in level of energy present at each level. Okay, in the base level, there is maximum amount of energy present because at this level, the energy is produced. As we move up, there is consumption of energy. That is why the amount of energy present at the upper levels is decreasing continuously. Okay, and what is the amount of energy produced at each level? It is 10% of the before level. So for example, this is level 1. And if you want to find out what will be the energy present at level 2, it will be 10% of the energy present at level 1. Okay, the energy present at level 2 will be 10% of the energy present at level 1. Now let us suppose we want to find out the energy present at level 3. What it will be? It will be 10% of the energy present at level 2. Understood? So that is how the pyramid of energy is broad at the base and narrow at the apex, because in the subsequent levels, the amount of energy that is present is decreasing because the energy is consumed by the organisms present at that level. Okay, understood? I hope it is clear to all of you. This, now let us see statement 2. It is saying the species that induce a bare area are called pioneer species. Yes, it is correct. A bare area does not have any population. The species of organisms, that reside in that bare area for the first time are known as pioneer species. So both the statements are correct and answer would be option number 4. Both statement 1 and 2 are correct. Now let us see question 105. It says if 20 joule of energy is trapped at producer level, then how much energy will be available to peacock as food in following chain? Okay, so producer is here plant, it has 20 joules. At each level, we have 10% of the previous level of energy present at previous level. So in mice, 10% of the 20 joule will be present. 10% of the 20 joule will be present. So which will be 10 into 20 by 100, which will be 2 joules. Okay, it is 2 joules. In snake, 10% of 2 joules will be present, which is 2 into 10 by 100, which is 0.2 joules. Okay, in peacock, 10% of 0.2 joules will be present, which is 0.2 into 10 by 100, which is 0.2 by 10 is 0.02 joules. Okay, so answer will be 0.02 joules. That is option number 1. Understood? I hope this is clear to all of you. It is a very, very basic and simple maths. If you do not understand, please let me know in comments. I will clear it out for you. Okay. Moving on to question 106. Which of the following is not a functional unit of ecosystem? Yes. As I told you, standing state includes functional characteristics of ecosystem, which includes species, population size, etc. So that is what it is asking. Which of the following is not a functional unit of ecosystem? So answer to this is stratification. Stratification level refers to vertical organization of species in different levels. Okay, it is not a functional unit of ecosystem. Okay, functional unit of ecosystem refers to component of ecosystem which performs different functions of that ecosystem. Okay, so a functional unit of ecosystem has to be component of ecosystem which performs different functions of that ecosystem. And stratification is a process, okay, is a term. It is not a functional unit of ecosystem. That is why option 4 is the correct answer. Rest all options are correct, which is energy flow. Yes, in an ecosystem, energy flow occurs from level 1 to 2 to 3. Understood? That is why it is a functional unit. Decomposition of organism of different decomposition of the organisms occur. Yes, it is a functional. Productivity refers to, yes, net primary productivity and gross primary productivity. That is amount of energy captured and stored through photosynthesis by plants or autotrophs. Understood? So option 106 has answer 4. Question moving on to question 107. What does it say? It says column 1 match the following. Column 1, column 2. Okay, so we have different organisms and areas. Okay, we have different organisms and areas. Where can we? Yes, so let us see the first organism. The first organism is quagga, which is seen in Australia. Next organism is dodo, which is seen in Mauritius. Stellar sea cow. This is in Russia. Very fun thing I want to say you. If you do not read properly, then you will go to Russia and maybe stellar sea cow will make some more sense to you then. I know, bad joke. Very bad joke. Thylacine is related to Australia. Okay, I'm sorry, quagga was Africa. Let me make a very simple thing for you. Okay. Yes. Just a second, just a second, just a second, just a second. Yes. See, quagga, Africa, dodo, Mauritius, stellar sea cow, Russia, thylacine, Australia. So here answer would be option 3. That is 1 is B, 2 is A, 2 is A, 3 is D. Yes, thylacine is 4 is C. So option number 4, sorry. I'm really sorry. One is A. Sorry, one is B. Quagga is Africa. One is B. Dodo is Mauritius, D. Stellar sea cow is Russia, that is A. And thylacine is Australia, C. So we have 1B, 1B, yes, 2D, yes, 3A, 4C. Third option is the correct answer. Okay. Then we have question 108. It says regression coefficient value for entire continent is, okay, regression coefficient value of an entire continent 0.6 to 1. Understood. So answer to question 108 is option 2. Now I know that all of you will ask me what is regression coefficient, madam, please explain. So let me explain it to you in very simple terms. Okay. So let us take a ecosystem. Okay, let us take a ecosystem. It has factors like ecological factors, like it has ecological variables. Okay, ecosystem which has variables like, let's suppose, organism and temperature. Okay. Now we want to determine relationship between this organism of this ecosystem and temperature of this ecosystem. So we will plot a graph. Okay, we will plot a graph, and this graph will help us determine relationship between the organism of this ecosystem and temperature of this ecosystem. Now using this graph, we will find out a value, a numerical value, which is using this graph, we will find out a numerical value which will be known as coefficient of regression or regression coefficient. Okay. So regression coefficient is a numerical value which helps us to determine relationship between two variables of an ecosystem. For example, organisms and temperature. Okay. Now, if regression coefficient is 1, it means that both the variables are related to each other and both the variables will increase. If one variable is increasing, the other variable will also increase. If the regression coefficient is less than 1, it will indicate that both the variables are related to each other. But if one variable is increasing, the other variable will decrease. If it is 0, that is, if coefficient of regression is 0, it will indicate that both the variables are not related to each other. For example, we take organism and temperature. So if the coefficient of regression is 1, or more than 1, it indicates 1 or more than 1. It indicates that the temperature and organism are related to each other, and organism will increase when the temperature increases. Okay. And if the coefficient of regression, let's suppose, is less than 1 for organism and temperature, it will indicate that the organism and temperature are related to each other, and organism will decrease if the temperature increases, or organism will increase if the organism will increase if the temperature decreases, or organism will decrease if the temperature increases. Understood? Now, if the coefficient of regression is 0 for organism and temperature, it will indicate that the organism and temperature are not related to each other. Understood? So that is coefficient of regression. So it is 0.6 to 1.2 for an entire continent. Now, question 109. This is total by hearting question. If you should just remember this. Okay, there's no concept here. So it means that the Earth Summit Historic Convention to Biological Diversity was held in, yes, Rio de Janeiro in 1992. Okay. Question. This is also a totally by hearting question, and it is very easy. Rivette-Popper hypothesis was given by Paul Ehrlich. Okay, Rivette-Popper hypothesis was given by Paul Ehrlich. So moving on to question 111, what does it say? Match the following. Geitonogamy then produce a short seed set even in absence of pollinators. Xenogamy, chasmoglemus flowers, and cleistogamous flowers. Okay, so let us see. Geitonogamy. Let us start from here because there's so much information on this page. We'll start it from here. What it is saying? What it is saying? It is saying cleistogamous flowers. Okay. So what are cleistogamous flowers? They are open flowers. What do they produce? Assured seed set in absence of pollinators. Yes. Chasmogamous flowers, what similar to flowers of other species with exposed anthers and stigma. Yes. Xenogamy, transfer of pollens, grains from anther to stigma of another flower. Okay. Geitonogamy, produce assured seed set. So see, first we will see, to solve these kind of questions, there's only one technique. You see one option, geitonogamy. What is geitonogamy? Geitonogamy refers to pollination between different flowers of same plant. Different flowers of same plant, isn't it? So it is, let us see if this is matching with it or not. Produce a short seed set even in absence of pollinators. Okay, we'll see. Transfer of pollen. See, geitonogamy requires pollinators, and xenogamy also requires pollinators. What does not require pollinators is self-pollination. Okay, autogamy does not require pollinators, but geitonogamy and xenogamy require pollinators. That is why produce a short seed set even in absence of pollinators is a wrong option, because geitonogamy requires pollinators. And what is geitonogamy? Geitonogamy refers to pollination between different flowers of same plant. So option 1 is not correct because it requires pollinators. Now let us see option 2. Transfer of pollen grains from anther to stigma of another flower of the same plant. Yes, this is geitonogamy. So this is option A. Okay, gytonogamy is transfer of pollen grain from the anther to the stigma of another flower of the same plant. Now, what is xenogamy? Let us see, what do we know? We know that xenogamy is pollination between different flowers of different plants, and xenogamy requires pollinators. Okay, so let us see, produce assured seed set even in absence of pollinators is a wrong statement because xenogamy requires pollinators. Now let us see third one, transfer of pollen grains from anther to stigma of different plant. Yes, this is xenogamy. So B is the answer for statement number 3. Let us see, what are chasmogamous flowers? Chasmogamous flowers are Chasmogamous flowers are okay. So chasmogamous flowers are open flowers. Means chasmogamous flowers have exposed stigma and exposed anther. Okay. So anther and stigma of the chasmogamous flowers are exposed because the flower has opened. Now flower is in bud condition like this. It has inside it has a pistil. Okay, inside it has a pistil and a stamen, and it is closed. So if the flower is closed, you can't see the pistil and stamen inside it, right? But if the flower is open, you will be able to see the pistil and the stamen, isn't it? Are you getting me? That is, stigma is a part of pistil and anther is a part of stamen. So if you are able to see pistil and stamen, you'll obviously be able to see stigma and anther also. So if the flower is open, then only you can see the contents inside it. If the flower is closed, you can't see. That is why chasmogamous flowers are open and you can see the exposed stigma and anther. So answer to this will be similar flowers of other species with exposed anthers and stigma. Okay, so the exposed anthers and stigma are chasmogamous flowers. So this is D. Okay, now what are cleistogamous flowers? They're closed flowers. Okay, the flowers do not open. They're closed in bud condition only. They are present. So if the flower is not opening, okay, if the flower is not opening, pollination will be autogamous. Why? Because if you want pollen grain from the other flower to come and land on the stigma of this flower, you need to expose stigma. Okay. If the stigma is not exposed, pollen grain might come from the other flower, but it can't land on the stigma only because flower is closing the stigma. Okay, so you need an exposed stigma for cross-pollination to occur. If the stigma is exposed, then only pollen grain from the other flower will come and land on the stigma. Okay, if the stigma is not exposed only, then there cannot be cross-pollination. Okay, understood. And what is cross-pollination? Cross-pollination is pollination between two different flowers. Okay, understood. So cross-pollination cannot occur in cleistogamous or closed flowers because stigma is not exposed. So what happens? The stigma is present inside this flower. And if pollination has to occur, what pollen grain will come? The pollen grain which is of this flower only. And so now inside this flower, we have a pistil and a stamen. Okay, so the stamen of this flower will produce pollen grains which will go and lie on the stigma of the pistil. So this type of pollination which is occurring is known as autogamous pollination. In this, what is happening? The pollen grain of the same flower of the same plant will pollinate the pistil of the same flower of the same plant. Understood? And inside this, there is no requirement of a pollinator. Okay, you don't want someone to take pollen grain from somewhere and put here because everything is happening inside this closed flower only. Don't require a pollinator. Understood? So that is why cleistogamous flowers produce seed set in absence of pollinators also because they do not require pollinators in the first place. So this is D. Okay, so it will be A2, B3, C4 and D1. A2, B3, C4 and D1. Option 2 is the correct answer to question number 111. Now let us see question number 112. What it is saying, a typical angiosperm anther is Okay. A typical angiosperm anther is bilobed. The anther is bilobed with two theca. That is, they are dithecous. Two theca means dithecous. Okay, this is very simple. We know the anther of the angiosperms is having two lobes and it is having two theca like this. That is why it is bilobed and dithecous. Okay. Moving on to question 113. What does it say? Dash generally differentiate a single megaspore mother cell in the dash of the nucleus. Yes. So answer to question 113 would be option number, let us see. First we'll see. Dash generally differentiates a single megaspore mother cell in the dash of the nucleus. So this is nothing but ovules. Generally differentiate megaspore mother cell. Okay. In where are megaspore mother cell present? Micropylar of the region of the nucleus. Okay. So answer to question 113 is ovules differentiate. See, megaspore mother cell is present in ovule, which will form embryo sac. And it is present toward micropylar end. Okay, so ovules generally differentiate a single megaspore mother cell. So what does it mean? In an ovule, there are multiple megaspore mother cells formed. Out of this, only one megaspore mother cell will get differentiated and it will form embryo sac. And this whole structure is present towards micropylar end of the ovule. Understood? Simple, no complication. So answer is option number 2. Sorry, I'll take it properly. Option 2. Okay. I know sometimes my ticking skills are not so good. Question 114. What type of pollination takes place in Wellisneria? See, direct statement ever from NCERT. ठीक है, NCERT को खोलो, उसमें डायरेक्ट वेलिस्नेरिया प्लांट देखो, उसमें टाइप ऑफ पॉलिनेशन देखो, उसको याद रखो, यहाँ पे आंसर दो. There is no concept here. It is direct statement. What type of pollinations is taking place in Wellisneria? Water pollination. Okay, water is the pollinator. So male flowers are carried by water currents to female flowers at the surface. Water pollination. Okay. Now see, there's one more very important thing here which we should remember. In Wellisneria, water pollination is occurring. Okay, we should remember that is okay. But inside the water, no. On the surface of the water, yes. So this is a very important point which your NCERT says and which you should remember that Wellisneria is an example of water pollination, and the pollination occurs on the surface of the water and not inside the water. Understood? Understood. I hope it is clear. Okay. Okay. Now, why is the pollination occurring on the surface of the water? Because the stalk of the Wellisneria is very long. So what happens is flowers are coming on the surface of the water. Understood? Now, moving on to question 115. What is the assertion and reason? UV radiation is a mutagen that induces mutations in organisms. Yes, very, very correct statement. It is saying UV radiation is a mutagen and it induces mutations. Yes, it is correct. It is a why UV radiation is a mutagen? Because it induces UV radiation is a mutagen because it induces mutations. Now, UV radiation is an ionizing radiation. What does it mean? Whenever it hits an organism, it ionizes the particles present inside that organism. That is why UV radiation can induce mutation. Understood? Now, What is the reason? Mutation is a phenomenon that results in alteration of DNA sequences leading to changes in genotype and phenotype of an organism. Okay, so it is saying mutation is a phenomenon that results in alteration of DNA sequences leading to changes in genotype and phenotype of organism. This is the definition of mutation. Yes, what is mutation? It results in change in the DNA, which will be present in the genotype, that is in the genes also, and in the phenotype, that is it will be visible to the people also. So this is the correct statement. Both one and that is assertion and reasons are correct. Now we have to see whether the assertion is correctly explaining the reason, or sorry, whether the reason is correctly explaining the assertion or not. Okay, so assertion is saying UV radiation is a mutagen that induces mutation. Because now, whenever you want to solve such a type of question, you should insert a because here. When you insert a because here and read this statement after that, you will understand whether this is explaining it correctly or not. Okay, so it is saying UV radiation. It is saying UV radiation is a mutagen that induces mutations in organisms because mutation is a phenomenon that results in alteration of DNA sequence leading to change in genotype and phenotype of organism. Is it correct? Tell me. Is it correct? Are UV radiations mutagens because mutation is a alteration in DNA sequence? No, right? Think again. Read. Yes. So we will read it again. UV radiation is a mutagen that induces mutations. Okay. Because mutation is a phenomenon that results in alteration of DNA sequences leading to changes in genotype and phenotype of organism. Yes. Now we are saying that UV radiation is a mutation. Why? Because it is inducing mutation. Now what is mutation? It is a phenomenon resulting in change in DNA or genes of an organism. It can be seen in the genotype and also on the appearances. Yes, the reason is correct. Okay. Why? Because assertion is saying that UV radiation is a mutagen inducing mutation. Reason is saying what is mutation? If this is mutation, UV radiation is inducing that mutation. Okay. So this is the correct statement. अगर हिंदी में बोलेंगे तो ये बोल रहा है कि UV रेडिएशन एक म्यूटेजन है जो म्यूटेशन इंड्यूस करता है. क्यों? Because mutation क्या है? Change in DNA sequence. So this is a correct statement. So assertion is correct and reason is also correct, and reason is correctly explaining the assertion. So assertion and reason are correct, but the reason is correctly explaining the assertion. That is option number 4 is the correct answer to question 115. Now moving on to question 116. Let us see what it is saying. Question 116 says, match the contrasting traits studied by Mendel in a pea plant. Match the contrasting traits studied by Mendel in a pea plant. Okay. So first is flower position. And here we have inflated, constricted. Is flower position ever inflated and constricted? No. Flower position is axial or terminal. So this is A. Okay, seed color. Seed color is yellow, green. Seed color is seed color yellow or green? Green, yellow. Okay. So now, let us see. Okay, flower position. What is the flower position given by Mendel or studied by Mendel? It is axial or terminal flower position. Okay, now coming to seed color. Seed color. What is the seed? Okay, seed color. Yeah, let us see seed color. What is the seed color studied by Mendel? Seed color studied by Mendel is yellow or green. Okay, seed color studied by Mendel is yellow or green. Now, what is pod color studied by Mendel? Pod color studied by Mendel is green or yellow. Okay, what is pod shape studied by Mendel? It is nothing but constricted and inflated. Okay. So we have A is 2. A is 2. Okay. A is 2. Then B, pod color. Pod color is green or yellow. B is 4. Okay. Then seed color. What is seed color? Yellow or green. Then pod shape. Inflated or constricted. That is 1. So Option number 3 is the correct answer to question number 116. Okay, moving on to 117. Mendel selected pea plant varieties as pairs which are similar except for dash true breeding with dash traits. Okay, dash true breeding with dash traits. So if it is, it is very simple. So there are some true breeding plants which have some traits. See, I'm sorry. Mendel selected pea plant varieties in which selected pea plant varieties. How many? 14. So here we'll write 14. Okay. As pea plant varieties as pairs which were similar. So all the 14 pea plant varieties were present in pair which were similar except for one pair which had contrasting traits. Okay. The answer would be option 2. Option 2. Okay. Question 118. Mendel proposed two general rules to consolidate his understanding of inheritance and monohybrid cross, law of segregation, law of dominance. Today, these rules are called the first law of segregation and the second law of the first one is law of dominance and the second one is law of segregation. Okay, so answer to question 118 is option number 1. Very simple question. This is very simple question. Very simple question. Okay, extremely simple question. Don't have to see the other options also. Now, question number 1119. Just a second. I'll erase this. Yes. The united knowledge of chromosomal segregation with dash and called it as dash. Okay, this is a very interesting question. Who united knowledge of chromosomal segregation? Who united knowledge of chromosomal segregation? Sutton. Sutton united knowledge of chromosomal segregation with Mendelian principles and called it as chromosomal theory of inheritance. So Mendel gave theory of inheritance, that how are genes transferred. Some genes are dominant, some genes have alleles, some are dominant, some are recessive. How are they transferred from one parent to children? All this is nothing but principles of inheritance, and this was given by Mendel. Now Sutton, what did he do? He applied this to chromosomes and called it as chromosomal theory of inheritance. So answer to this question would be Sutton, because Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it as chromosomal theory of inheritance. Understood? Now next question, 120. The disease phenylketonuria, which occurs in humans. The disease phenylketonuria occurs in humans. The disease is caused by mutation in gene that codes for enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase converts phenylalanine, which is an amino acid present in your and mine body, to tyrosine. Okay, so if this enzyme is absent or mutated, phenylalanine cannot be converted to tyrosine. Understood? I hope this is clear. Very simple question this is. I think my eraser is not working. I have to find an eraser. Yes. So answer to question 120 is third option. Okay, I'm taking care and taking the answers. I'm sorry. I'm taking care and taking the answers correctly. Okay. The genotypes of a husband and wife are IA, IB, IA, small I. Among the blood types of the children, how many different genotypes and phenotypes are present? Okay, so let us take IA, IB, IA, I small. Okay, so this will form a list like IA, IB, IA, small I. Okay. Okay, understood. So now let us make a Punnett square. IA, IA, IB. Okay. IA, small I, IB, IB, small I. So we have IA. So as we can see, what are we getting here? We are getting IA, IB, IB, small IA, small I. So we are getting 4 genotypes. All these are different genotypes. And this will be A blood group, this will be AB blood group, this will be B blood group, this will be A blood group. So these two are same. Both these are A blood groups. So this is A blood groups. So we are getting 1, 2, and 3. 3 phenotypes. So 4 genotypes, 3 phenotypes. So answer would be 4 genotypes, 3 phenotypes. Second option. Okay. Moving on to 122. Purines found both in DNA and RNA are, yes. Now see, if you don't know this question also, I suggest please open your NCERT and open biomolecules chapter of your class 11. Okay, because if you don't know this also, I don't know what blunder are you going to make in NEET examination. Okay, so coming to this. Purines found in both DNA and RNA are adenine and guanine. Pyrimidines are thymine, cytosine, uritin. Okay. 123. The 3-5- phosphodiester linkage inside polynucleotide chain serve to join. Okay. So the 3' to 5' phosphodiester linkage inside a polynucleotide chain serves to join. What it is asking? Nothing it is asking. It is asking that 3' to 5' phosphodiester bond in a DNA is formed between what? One DNA strand and another DNA strand. I don't know. Or one nucleoside to another nucleoside, one nucleotide with another nucleotide. See, very simple. Phosphodiester bonds are present inside DNA, right? So these cannot link two DNAs. No. Phosphodiester bonds are present inside DNA. So phosphodiester bond definitely cannot link two DNAs. So this statement is wrong. First statement is wrong. Okay. This question is asking 3' to 5' phosphodiester linkages are present inside a polynucleotide chain which serve to join DNA molecules. No, they are present inside the DNA. How will they join DNA? Not possible. One nucleoside with one nucleoside? No. No. Phosphodiester bonds are present between phosphate molecules. Okay. And if at all there is phosphate molecule present, it means it's a nucleotide. Okay. What's a nucleotide? It's a ribose, 5-carbon ribose sugar. Okay. One nitrogen base and phosphate. Understood? If this particular structure is present, it means it's a nucleotide. And what is the identification feature of a nucleotide? Phosphate. Why? Because if this 5-carbon sugar with nitrogen base is present, then it will be a nucleoside. So you are adding a phosphate to a nucleoside to form it a nucleotide. Understood? So phosphodiester bonds are formed between phosphates. Okay, and if phosphate is present, it means that it is a nucleotide. Okay, so phosphodiester bonds are joining nucleotides to form polynucleotide chain. Okay, understood? Understood now? It's a very easy question. Answer to this question is option number 3, which is one nucleotide with another nucleotide. See, phosphod. Understood. If at all you don't know so much DNA structure, I'll tell you one simple trick. What it is saying, 3' to 5' phosphodiester linkages inside a polynucleotide chain serve to join. Polynucleotide chain means you will have nucleotides. No? So what it will serve to join? One nucleotide to another nucleotide. Understood? Third option is the correct answer. Okay. Nucleosome core is made up of, yes, H2A, H2B, H3, and H4, and not H1. H1 is absent in nucleosome. Okay. 125. Unequivocal proof of DNA as the genetic material came from studies of. I know you were thinking. I know you were thinking Hershey and Chase, but I think unequivocal proof of DNA came from studies of bacterial virus, also known as bacteriophage. Now, why they have given bacterial virus? Because they want to test you that bacterial virus is also called bacteriophage, because bacteriophage is written in NCERT. Some people will blindly read bacteriophage, remember it. Nobody will know that it is a bacterial virus. That's why they want to test you whether do you know that bacteriophage is a bacterial virus or not. Okay. 126. Similarity in DNA and RNA is that, see, this is a very simple question. Okay. Both are polymers of nucleotides. Both have similar pyridines. What are pyridines? Come on. Cytosine, thymine, urine, uracil. So, are DNA and RNA having similar pyrimidines? No. So wrong. Both have similar sugar. No. One is having deoxyribose. One is having ribose. Wrong. Both are genetic material. Okay. Are both genetic materials? Come on. Is RNA transferred for one person, one parent to another children? No, right? Can RNA be transferred from parent to children? No. So it's also understood. Answer to question 126 is option number 1. One 27. The special protein that help to open up DNA double helix in front of replication fork. The proteins are, so they want to decoil the DNA strand. And decoiling of DNA strand happens with topoisomerase. DNA topoisomerase. So DNA, all this, if you do not know also, you should know topoisomerase is an enzyme which opens to which opens the DNA or helps to uncoil the DNA. Okay. 128. AGG, TAT, CAT is a sequence from coding strand of a gene. What will be the corresponding sequence of transcribed mRNA? mRNA and coding strand have same sequence, except that in place of T, you have U. So you will have A, G, G, U, A, U, C, G, C, A, U. So answer is nothing but A, G, G, U, A, U, U, A, U, C, G, C, A, U. Yes, that is option number 1. 129. Which of the following pair of codon is correctly matched with their function or the signal for the particular amino acid? AUG, ACG is a start codon, codes for methionine. Okay, it's a correct statement. Okay, correct statement. AUG is a start codon. ACG do not code for methionine, so this is wrong. Okay, understood. This is wrong. UUA, UCA codes for leucine. UAA, ochre, umbel. UAA is an ochre. Okay. Ochre, UAA is ochre. And it's a stop codon. But this is UUA and UCA. Okay, UUA and UCA code for leucine. No, this is wrong. GUU and GCU code for aniline. This is also wrong. UAG, UGA. UAG, UGA, and UAA are stop codons. Yes, it's a correct statement. UAA, UAG, UGA, also known as ochre, amber, opal. Stop codons, they signal to stop the protein synthesis. Okay, so if at all you don't know that this is coding for leucine, this is coding for this is so definitely wrong. AUG is a start codon. ACG does not code for methionine. So this is wrong. Okay. If at all you do not know that UAA, UCA are coding for leucine, GUU, GCU are coding for alanine, but you know that fourth option is correct, right? Matlab, this can be correct or cannot be correct. This can be correct or cannot be correct. But this is definitely correct because we know. So answer to this question is fourth option. See, I'll tell you one trick to solve MCQs. You cannot always know all the options. Okay, so you have to follow the rule of elimination. Eliminate the most wrong options, and then when you are left with the most correct options, use your brain. Okay. Protein synthesis in an animal cell occurs only on ribosomes present in cytosol, only on ribosomes attached to nuclear envelope and endoplasmic reticulum, on ribosome present in nucleus as well as in cytoplasm. Yes, we'll see. On ribosomes present in cytoplasm as well as mitochondria. Yes, so mitochondria is a semi-autonomous organelle. It has ribosomes which can synthesize proteins. Yes, this is correct. Ribosomes present in cytoplasm. Yes, this is also correct. So answer to this question is option number 4. And why not the other options? Because only ribosomes present in cytosol is wrong, because it can, ribosomes, if they are present anywhere, it can synthesize protein. So this is wrong. Only ribosomes attached to nuclear membrane and E are also wrong. I told you, if ribosome is present anywhere in the cell, it can synthesize protein. On ribosome present in nucleus and in cytoplasm, this is also wrong. Okay, because in nucleus, rRNA is synthesized. Ribosomal RNA. 131. One of the most frequently used techniques in DNA fingerprinting is, now see, this is such a simple question now. Such a simple question now. That matlab, you should not know anything. You should just know, if you have ever seen DNA fingerprinting topic also, in that one word will be there. That one word is the answer. Okay, what is it? VNTR, variable number of tandem repeats. What does VNTR stands for? Variable number of tandem repeats. Okay, what does it means? It means that a particular, let's say ATTCA is a gene sequence. It is repeated in a DNA variable number of times. Understood? So that is variable number of tandem repeats, and it is used as a most frequently used technique in DNA fingerprint. Question number 132. What does it say? It says, during formation of bread, it becomes porous due to release of carbon dioxide by action of yeast. Baker's yeast. Scientifically known as Saccharomyces cerevisiae. 133. Monascus purpureus is a yeast used commercially in production of, come on, it is very easy, blood cholesterol lowering statins. HMG coenzyme A inhibitors are known as blood cholesterol lowering agents, otherwise known as statins. Monascus purpureus is used in production of statins. Okay. 132. The citric acid is produced, 134. The citric acid is produced by Aspergillus niger fungus. Aspergillus niger fungus. What gases are produced in anaerobic sludge digesters? So yes, this is nothing but sewage treatment plant. And what it is asking, what gases are produced in anaerobic sludge digester? So answer is methane, hydrogen sulfide, and carbon dioxide. The type of gases produced in an anaerobic sludge digesters are methane, hydrogen sulfide, and carbon dioxide. Okay. Methane, hydrogen sulfide, and carbon dioxide. Understood? See, the anaerobic sludge digesters has methanogens, archaebacteria, yes. These produce methane plus carbon dioxide. Okay. And there are these bacteria, there are another anaerobic bacteria which can produce hydrogen sulfide. Apart from these three gases, which are very important, sometimes ammonia can also be seen. Okay, I hope this is clear to you. Moving on to question 136, it's an assertion reason type question. It says majority of insect pollinated flowers are large, colorful fragment rich in nectar. Yes, because we have to provide insects some reward for their pollination that they are performing for flowers, and that reward is nothing but nectar. And these large colorful fragment flowers are for attracting insects. Okay, because insects will come to flower only if it is attractive, right? So it has to be large, colorful, nicely looking fragment. So it's a correct statement. Animals are attracted to flowers by color or fragments. The flowers pollinated by flies, beetles secrete foul odors to attract these animals. Okay, this is a correct reason. Okay, so the both statement, assertion and reason are correct. Both assertion and reason are correct. Okay, so the statement we'll insert because here and read as I told you. It is saying majority of insect pollinated flowers are large, colorful, fragment and rich in nectar because animals are attracted to flowers by color or fragments. Okay, correct. The flowers pollinated by flies and beetles secrete foul odors to attract these animals. Now see, if at all this statement was not given, then it is both assertion and reason are true, and reason is the correct explanation to assertion. But this statement is given, right? And this statement is not in sync with what it is saying. It is saying that flowers which are pollinated by flies and beetles secrete foul odor. And here it is saying it is fragrant, and fragrance is nowhere similar to foul odor, right? So both of them are correct statements, but reason is not the correct explanation to assertion. Therefore, option number 2 is the correct answer to question 136. Okay. The process of formation of microspores from pollen mother cell through meiosis is called microsporogenesis. It's called microsporogenesis. Through the microspores as they are formed are arranged in cluster of 4 cells, and it is known as microspore tetrad. Option number 1 is the correct answer to question 137. Okay, 138. What does it say? It says below are two given statements about sex determination in honeybee. Male bees have half the number of chromosomes than that of the females. Okay, male bees have half the number of chromosomes than that of a female. Okay, that is true. In a honeybee, the sex determination in honeybee is based on number of sets of chromosome and individual receives. Yes, if half it is receiving, it is a male. If it is receiving full, then it will be a female. Yes, it's true. So both statement 1 and 2 are true. A gene showing codominance has alleles that are recessive to each other? No. Both alleles are independently expressed in heterozygote? Yes. One allele is dominant on other? No. Alleles are tightly linked on the same chromosome. Tightly linked on the same chromosome? No. Answer to this question is both alleles are independently expressed in a heterozygote. Okay. For example, for this codominance is AB blood group. IA and IB both are independently expressed, and they give a AB blood group. Moving on to 140. Statement says satellite DNA is important because, yes, why is satellite DNA important? Yes, we should know this. This is very important because it shows high degree of polymorphism in population and also the same degree of polymorphism in individual, which is heritable from parents to children. Okay, so answer to this question is option number 4. C. See, one thing I want to tell you about satellite DNA, it is very important. So, see, satellite DNA is nothing but a type of DNA which has repetitive repetitive sequence of genes. And how many times this particular sequence is repeated is variable in different individuals of population. Okay, so I'll give you an example. Let's suppose we have ATGC, ATGCA. This sequence is present. Okay, now if this sequence is repeated in a DNA of an organism, it will be known as satellite DNA. Okay, now how many times this sequence is repeated is variable. It will depend on different organisms. Okay, different individuals will have different number of repetitions. And how long? See, this ATGCAA is this long. There can be a sequence which is CGCCGCAA. Now this is longer than this, right? So this sequence, my friends, is a microsatellite. This is a mini satellite. So depending on the length of the satellite DNA, they are classified into micro and mini satellites. Okay, so satellite DNA, one benefit of satellite DNA is that the number of times it is repeated is variable in different individuals of different populations. Means every organism has a different number of repeats of satellite DNA. Okay, and this helps in identifying that organism. And it is used in DNA fingerprinting. Okay, so the importance of satellite DNA is that only. Okay, and this particular nature of satellite DNA, where it is repeated variable number of times in different organisms, is known as polymorphism. Okay, so what is polymorphism? Polymorphism refers to variable number of repeats of satellite DNA in different organisms of different populations. Okay, so every organism has a different number of repeats of satellite DNA which is unique to that organism, and this is known as polymorphism. Show high degree of satellite DNA shows high degree of polymorphism in population, and also the same degree of polymorphism in organisms. Okay, understood. And this polymorphism, that is this the number of repeats, how many times the satellite DNA will get repeated, will be transferred to parent to children. It is heritable. Okay, so now question 141, the lac operon consists of one. Answer to question 141 is one regulatory gene and three structural genes. Okay, I, O, I, Z, Y. I, Z, Y. I is inducible. Lac operon consists of one regulatory gene and three structural genes, which are Z, Y, Zaya. 142. Amino acid sequence in protein synthesis is decided by sequence of mRNA. Okay. So what are different codons present on mRNA decide the sequence of amino acids in a protein? Okay. Mycorrhiza exhibits phenomenon of symbiosis. Mycorrhiza is a symbiotic relationship between fungi and algae, wherein both fungi and algae are benefited. One gets shelter, one gets food. Fungi gets food, algae gets shelter. A high density of elephant population in an area can result in, this is a very simple question. High density of elephant population in an area can result in intra-specific competition. See, if there are so many human beings writing NEET, I'll give an example. Okay, there are 25 lakh aspirants which are writing NEET UG examination this year. And all of us are human beings. Nobody is an elephant, right? All of us are human beings. So there's a competition between human beings, and all of human beings are same species. So this is known as intra-specific competition. Okay, what is intra-specific competition? Competition occurring among same species organism or organism of same species. So if so many elephants are there in one population or in one area, then there will be competition between elephants, which will lead to intra-specific competition. 145. A prokaryotic autotrophic nitrogen fixing symbiont is found in. Yes, this is such a simple question. Cycas exhibits relationship with symbiotic relationship with nitrogen fixing bacteria. Okay. Okay, I want to tell one little thing here. Just a second. Mycorrhiza, I'm sorry for my mistake. Sometimes out of flow of teaching, some things happen. Mycorrhiza is a symbiotic relationship. What is a symbiotic relationship? It is a plus-plus relationship wherein both the organisms are benefited in one or other way. Okay. It is a relationship present between roots of higher plants and roots of higher plants and fungi. Okay. Not in algae or fungi, not algae and fungi. The relationship between algae and fungi is also symbiosis, which is known as lichen, not mycorrhiza. So cycas is a symbiotic relationship between nitrogen fixing bacteria and the plant itself. The domestic sewage in large cities has, yes, high biological oxygen demand as it contains both aerobic and anaerobic bacteria, is processed by aerobic and then anaerobic bacteria in secondary treatment in sewage treatment plant. When treated in STP, does not really require aeration step as sewage contains adequate oxygen, has very high amount of suspended solids and dissolved liquids. Yes. So this is a very simple question. Okay. It is a domestic sewage in large cities is processed by aerobic and then anaerobic bacteria in secondary treatment plant of STP. Okay, so answer to question 146 is option number 2. Okay. One 147. Principle of competitive exclusion was given by Gauss. Principle of competitive exclusion was given by Gauss, GF Gauss. Which of the following is categorized as protein in true sense? Which of the following is categorized as protein in true sense? Female Anopheles bites and sucks blood from humans. Sorry, which of the following is categorized as parasite? That's what I was thinking. I read out protein somewhere. Parasite. Okay, so what is a parasite? A parasite is the organism which resides on or in another organism, which is known as a host, and it derives nutrition from the host. So female Anopheles bites and sucks blood from the humans, yes, but it does not reside on humans, right? So female Anopheles mosquito cannot be a parasite because it does not reside on humans. Human fetus developing inside a uterus draws nourishment from the mother. Okay. Head louse living on the human scalp as well as laying eggs on the human air. Okay, so head louse is living on us and deriving nutrition from us. So that's why it's a parasite. Correct. In true sense, the cuckoo lays its eggs in crow's nest. It's not a parasite, right? Because no. Because even though the cuckoo has left its egg for residing in the nest is not on the body of crow, right? It has to be, the parasite has to be residing on or in the body of the host. Nest, the cuckoo's eggs are residing in the nest and not on the body, right? That's why this is also not a parasite. Now, human fetus is residing inside the uterus and is taking the nourishment from the mother, but it is not a parasite in true sense. Okay, it's not a parasite in true sense. That's why answer to question 148 is option number 3, which is head lice. Head lice, they live on our scalp and derive nutrition from our blood, from the blood vessels in the scalp. So, I'll answer to question 148 is option number 3. Let us see 149, the relationship between algae and fungi is lichen, which is a symbiosis. Yes, I told you, right? In before question. 150. Which type of association is found between Entomophilus flower and pollinating agent? Yes, it is a mutualism. Both of them are benefited. Okay, both of them are benefited. Mutualism is plus relationship where both of them are benefited. Commensalism is plus and zero relationship, whereas one organism is benefited. There is no effect on the other organism. Cooperation is not, cooperation and co-evolution are not a type of species interaction. Okay. So this was about the answers to questions of your part test 4. I hope it was clear. If you have any doubt, you can mention in comments. I'll happy to clear them. I'm very happy to clear them. Also, this zoology section will be taught to you by your Sakshi ma'am. So wait for her. See you with the next video. Bye.